Good evening, everybody. Usually I stand by the light switch to do this. I'm not by the light switch. Well, welcome, everybody. We're glad you're here. My name's Lad Akins. I'm the Director of Special Projects at REEF. And this is REEF's monthly open house we call Fish and Friends. So every month we do this, the second Tuesday of each month. And we open up next door, refreshments, a little bit of snacks, and then we come over here and have a guest presentation. Before we get started, there are a couple of quick things. We always do announcements. The most important announcement is that there's a white Virginia SUV with Virginia plates outside. The alarm's going off. So maybe somebody tried to break in. Probably not.
Somebody tried to raffle it off.
The Virginia plates that did it.
So before we get started, how many people are here for the first time at our open house? Welcome. You have been here before. We have some ringers. Somebody.
You buy the beer.
This is a free event that we put on each month, and thanks to all the volunteers that have brought hors d'oeuvres for us. We really appreciate that. We're going to do a couple quick community announcements before we get started. First of all, coming up soon, Carlos and Allison and Estaffe are going to have their 100 Fish ID seminar January 25th in this building. 7 p.m. There you go. That's right. Also, this coming weekend at Founders Park in Islamorada is the Bluegrass Festival. And we're going to have a booth there on Sunday. So feel free to come by and share some bluegrass and activities. The Lionfish Derby series for the summer has been set. But we also do a winter derby each year. And that's primarily for people that are locals, even though it's expanding each year. The date for the Winter Derby is March 31st. So the last Saturday in March. So if you're interested in that, put that on the calendar. Are there other community announcements? Does anybody have anything they need to throw out? Any CRF announcements?
Is the Fish and R the same date as, is the 25th, right? There, I saw it on the.
January 25th is
That's your, that's,
Fish.
that's a,
ID class.
yeah, but there's a, I saw it in the email called the Fish and R.
I'll check for you and get back to you on that.
Fish and ours up
Yep.
online.
Yeah, that's on.
He's online, yeah. So they're, I think they're opposite,
All right.
same day.
Well, I'll get you the answer. All right. So, last but not least,
That's a good point.
last but not least, this is our brand new interpretive center. We are really excited. And so please take time to explore, look around, read the signage, and we're welcome to comments and things we can do better here, as always. All right, I'm going to turn this over to Nancy. You wanna
To
do
the
this?
2017 volunteers.
Yes, the Volunteer of the Year, Miss Nancy Barrett.
Oh, this is going to be interesting. Can you hear me all right here?
Yeah.
First of all, thank you so much for coming. We really appreciate your support, and I'm sure you're going to enjoy this new interpretive center. We're very, very proud of it. All of these photographs were donated to us by Stephen Frank, and they're all local with the exception of one. The pillar coral is from Cuba. And also the diver up there happens to be his daughter. That's a lot. Anyway, we're very excited that Margaret Miller is our guest speaker tonight. She's the research director for SECOR. I knew I was going to have a problem with this. For SECOR International, which is a conservation nonprofit dedicated to larval restoration of corals. She received an undergraduate degree from Indiana University and a doctorate in marine ecology from the University of North Carolina at Chapel Hill. Her dissertation involved ecological studies of non-reef building coral, Oculina spp, off North Carolina. She then moved south to examine some real coral in a 3-year postdoctoral position with the University of Miami. She spent 19 years as a research ecologist with NOAA Fisheries Southeast Science Center, leading an active field research program in the Florida Keys focused on coral early life history, coral restoration, population studies of threatened elkhorn and staghorn corals and their threats. She resides in Miami, Florida with her husband and 14-year-old son. And welcome. We really are so pleased.
Thank you so much. I have been looking forward to coming down and speaking with Reef for some months. Ellie and I have been trying to make this happen since way back before Reef Fest, and then when Reef Fest got postponed, and we've been trying to reschedule. So I'm really thrilled to be here, and it's particularly exciting to be in this great new facility. So that's a great privilege for me. As Nancy mentioned in the introduction, during this time, since I originally agreed with Ellie to speak for Reef, I've actually changed jobs. So she didn't get the speaker that she originally bargained for exactly. But I've tried in this presentation to blend a little bit of my previous work with NOAA and the work that my new organization, SECOR International, is involved in throughout the Caribbean. That's one of the exciting things about my new position, is I'm really working across many areas in the Caribbean on coral restoration. So I hope you'll bear with me. I may do a little bit of jumping back and forth, but hopefully that won't be too confusing. And that way, all the bases are covered, as we say. Maybe that's not going to work. All right.
Wow.
So, you always have to give an introduction to the talk, but when I'm in the Keys, it always feels a little silly. So, you all know how important coral reefs are. You know why they're important. Tourism is obviously a huge aspect here in the Keys, fisheries. Coastal protection is an aspect of coral reefs that in a lot of areas now we're coming to appreciate a lot more and think about sort of assigning economic values to. As we have more storms and we become more concerned about things like sea level rise, this is a really important function of reefs in a lot of developing countries where that shoreline protection is incredibly valuable. So these are all important values that corals and coral reefs provide. And again, as economists attempt to put numbers, right, on the economic value of these resources, you get a lot of crazy numbers, but 30 billion per year was one that I had seen for worldwide coral reefs. On the other side of that coin, I know you're also all probably very familiar, especially here in the Keys, we've had drastic population declines of most corals, those living animals that provide that structure that builds the reef and provides that habitat and that coastal protection. And there is a long litany of reasons for that. The very obvious ones here recently, I don't know how many of you were diving in 2014 and 15 and saw sites like this. This is Grecian Rocks Reef, a formerly really gorgeous stand of elkhorn coral that completely died during the 2014 bleaching event. So there's a lot of things that are killing corals and making it hard for them to survive, but equally important is the fact that these reef-building corals particularly are not recruiting well, so we don't have new members joining the population. And both of these factors are really the driving factors. Both of them are important in terms of driving population decline. So this recruitment failure particularly is an area where coral restoration can directly address by jump-starting or circumventing some of those steps which are problematic in the recruitment process.
Is recruitment a diversity process, or is it is it just adding to the population?
So I'm talking about adding to the population, but there's a lot of aspects of the population. I'm gonna talk. Hold that thought. It's a good one.
Margaret, can I? There's something I forgot to mention in the beginning.
Sure.
We are broadcasting this remotely and next door, so you're mic'd up. So they're getting that. So any of the questions in the audience,
I
they're
should
not
repeat.
catching.
Thank you
Sorry
for
to
good
bring that
reminder.
up earlier.
Remind me if I forget, but yeah, good, thank you. But anyway, as a result of these population declines from a lot of these different factors, we actually have 7 local Caribbean species of coral that are listed under the Endangered Species Act. When I worked for NOAA, this was one of the processes that I was actually involved in, the process of evaluating coral species for listing under the Endangered Species Act, and some of the other management and recovery planning that is involved with ESA listing. Something that's interesting, we also have 15 Pacific coral species that were listed at the same time. But the Pacific species that are listed under ESA are sort of species you would expect to be endangered. The rare species, right? The ones that you never expect to see anyway. The difference with our Caribbean species that are listed is that 5 of the 7 are our core reef-building species. They are the most abundant species that are in fact threatened. So you see, that's a very different scenario when you think about the fate of the reef, when your foundation species are the ones, the super common important species are the ones that are really in trouble. So again, this points to some of the importance of restoration for these species. And so just another couple points about these foundation species of coral reefs. Many of you who are in the water will recognize these as, again, supposed to be the most common species on the reef, the elkhorn coral on the top, and Orbicella fabulata goes by mountainous star coral, mounting star coral on the bottom. These are the most important reef-building corals in our area historically. Now, not so much, right? But they share common ecological characteristics, particularly about their breeding systems, that sort of contributes to this recruitment failure question. And so they're both broadcast spawning species, and most Caribbean reef-building species are broadcast spawning. So that means all of their gametes are just spit into the water and have to make all of the developmental steps sort of on their own, starting from an individual cell, not even from a zygote. So you can imagine that's a challenging accomplishment in the open ocean. Spawning tends to be rare. It only happens once or twice a year. The gametes are actually short-lived. They have to fertilize within a couple of hours after they're spawned. So that means you need to have enough parents spawning at the same time that are different from each other. So this is the other thing. Many of them are hermaphrodites, but they're obligate outcrossers. So that means even though an individual parent is producing both eggs and sperm, they can't fertilize together. They have to have a distinct, genetically different parent with which to fertilize. And that means those genetically distinct parents have to be close enough on the reef that those gametes will float together. And they have to be able to spawn at the same time because the gametes are only going to last for a couple of hours. So you can see this is overall a challenging accomplishment in the open ocean. And as the adult populations decline, as we have fewer parents out there, it becomes more and exponentially more difficult. This is a process we call depensation. All right, and I have a definition here that's kind of a sciencey definition, so I apologize for that. But if you just think of this as the downward spiral, right? So when a population reaches a certain low level, it means that it's going to keep going in that direction because these reproductive processes become more and more and more challenging. And so our concern really, especially in the Florida Keys, is that several of our reef-building species are really approaching these types of processes where the natural reproductive processes are really substantially impaired because we don't have enough parents, enough different parents spawning at the same place and time to accomplish fertilization, enough fertilization then to make it through all the subsequent bottlenecks. So that's kind of the setup, right? That's the situation that we find ourselves in. And so what I was just describing, that process of sexual reproduction, is what I was just describing. Now corals, many types of corals, can also reproduce asexually, and especially for the elkhorn and staghorn corals, this is a huge advantage because you don't have to go through all those processes I was just talking about. If a little piece drops off or breaks off, those can form into new colonies. And much of our coral restoration effort and coral propagation effort has utilized this characteristic of corals to enable the propagation by fragments. And so this has been very successful, and it can be accomplished, at high levels and relatively short periods of time, because you can start with you can start with a baby essentially that's this big instead of starting with a baby that's microscopic, right? Which is what the larvae start as. However, when we use asexual or fragments for restoration, we're going to raise them and outplant them. Our goal is still that there is a sexually reproducing population at the end, right? Because that's what we need for a population to be sustainable in and of itself. So that's still a goal. That's just not the part that we're actively working with. We're restoring more adults, and we're assuming they're going to take care of that on their own. Yes?
So that statement assumes that, or infers that genetic diversity is important for...
That's exactly right.
As you repeat the question.
Yes. So the question, which was also asked earlier, is a good one. Is about genetic diversity and how that relates to effective recruitment and reproduction, right? And so yes, genetic diversity is important for a couple reasons. So one of them, as I mentioned, you have to have genetically distinct parents. And so for some particularly for fragmenting species like elkhorn and staghorn coral, a lot of times you may go out and see a lot of colonies, but you may only have one genetic parent. So even though you have a huge, beautiful, healthy-looking reef with lots of elkhorn coral, you may only have one parent, and that means no matter how many eggs and sperm they put out, you get 0 babies. Okay? So it's important in order to have enough parents, especially for clonal species. But it's also important, especially as we move into the future, for coping with a lot of the environmental changes that we know are going on, with warmer temperatures, other types of threats that have to do with the environment. In order for corals to be able to adapt, our understanding is that genetic diversity needs to be maintained and, if possible, increased, because that gives more opportunity for resilient and survivable corals to be a part of that population. So it is a very important step for maintaining and increasing genetic diversity in populations. Now, so then if you're doing restoration, you can also start with the sexual reproduction. As I said, there are challenges to this, and we're going to talk through some of those, but it has the advantage of enhancing this genetic diversity at the front end instead of at the back end, essentially. So the process is fairly different. I want to emphasize too, though, we're really looking more towards using hybrids between these two approaches in restoration as well. And so these are aspects such as using these in situ coral nurseries, such as Coral Restoration Foundation has amazing facilities and have been used a lot this way as brooding stocks. So we can collect spawn actually from the nursery corals. We have all those parents all in the same place, and that works really well. And that's a really important sort of hybrid approach to restoration that we're also thinking more about and implementing in more areas. But this sexual reproduction, larval reproduction, as I said, is the aspect that Sea Corps really focused their efforts and research. So we'll talk through some more of those operations. So as I mentioned at the beginning, I'm going to give a little bit of a mashup of some of my earlier work and some of my current work at Sea Corps. So in my earlier work, I did a fair amount of research in collaboration with the local coral nurseries that use fragmentation for propagation. Many of you are intimately familiar with this process, but this is just sort of an illustration of the steps for fragment-based culture, where small fragments are harvested from wild populations. They're cultured often in the ocean, in ocean nurseries, and then the cultured fragments are actually outplanted on the reef, either using epoxy or nails and cable ties or some way of attaching them onto the reef. And then again, here you see, hopefully, ultimately, those restored populations then are at a density, a diversity, and a health level that they are able to reproduce successfully on their own. So that's kind of the process of coral restoration via fragmentation, or coral gardening is a term that's often used for this approach. So this is an area that's had a lot of investment and success in the Florida area. There's a lot of different organizations. CRF is the primary one, but there's a lot of other organizations engaged in this type of activity. And again, on the diversity point, there's over 200 genotypes of staghorn coral that are in culture in Florida. And again, that's a very explicit part of the strategy to maintain as many genotypes as possible in this process for the reasons that we've talked about. It's even hard to keep up with the number here in terms of the totals outplanted. I don't know if you CRF people have updated. I mean, this is a few of the nurseries up to 16, but you know, it's very large numbers of corals that have been put out on the reef. Yes.
Did the organizations swap genotypes?
So that's a good question. I would say, so the question was if the different organizations running nurseries swap genotypes back and forth. And this has actually been a problematic question in the past, because as we described, although diversity is important, we also have an understanding from conservation genetics that often, or we might expect, that individual genotypes may be particularly suited from the location they came from. And so there has been a lot of reluctance about moving genotypes around too much. Now, that being said, there's also more recognition as well that more genotypes is maybe still is probably better. So there is some exchanges happening in adjacent nurseries, essentially, right? So there are nurseries spanning all the way from Broward County all the way down through the Dry Tortugas. And so adjacent nurseries will sometimes exchange genotypes, but I think that's pretty much the extent of it in terms of how operations are done now. And those are permitting restrictions where the state of Florida won't allow you to move them too much further than that. Mm-hmm.
There was a study done between coral restoration in the Florida Keys and Miami, was it? And you know, the studies were done with how did these corals
see how they were when you moved them that far.
They all did best at home.
Where they came from. So it's a difficult question. It's actually a pretty difficult scientific question. It's one that a lot of research is focused on. But in general, again, in doing all of this, the approach, the management approach has been to sort of, let's move forward, but let's do things in the least risky way that we can. And it was just deemed that that's an aspect where it might represent a risk, it might not, but in case it does, we don't need to move them around, and let's stick with keeping them as close to home as possible. And that's just been the general approach. So again, many outplanted sites, and this published paper that published last year, compiled the data from a bunch of different outplants and came up with an overall average about 85% survivorship over the entire region, including one nursery in Puerto Rico as well, of 85%. So, yes?
Is there any initial data on the survivorship on those outplants after the hurricane?
Yeah, I don't know that there is. I mean, it's obviously a concern. I know many of the nurseries took a lot of damage, so we know that a lot of the stocks in the nurseries. I know some of the outplant sites in Miami-Dade County were completely devastated. I'm sure there was damage, like the damage to the reef itself. I imagine it was fairly patchy. Some places were probably pretty well wiped out, and other places you wouldn't necessarily know that there was too much of a problem. But I don't think there's been a comprehensive data look post-hurricane. But you know, this is a pretty impressive set of numbers, I think. You know, we'd say, wow, that's pretty good. You know, it is true that when we outplant corals to the reef, they're still subject to those same list of things that we talked about at the beginning that cause corals to die. And so, you know, mortality still happens to outplants. That's sort of natural because that means they're acting like the wild colonies, which is kind of what we want them to do, right? So it's a little bit of a tough balancing act there. But, you know, there are critics to this process that would say, well, this is all well and good, but the cost is really high. And, you know, mortality is still happening. These numbers may look good, but I could also look at 85% survivorship in a year and say, well, that's really not so good because if you take that out 4 or 5 years, you don't end up with any corals, right? So there's a critique here, but a lot of it has to do with scale. The fact that we are outplanting what sounds like a pretty impressive number of corals, but also represents a very large amount of effort and investment. And so, is there really ever any hope that we could make a real dent on the reef scale with this type of activity? So, I mean, another way to think about this, right, like you know, we quote the number of fragments, but we should really be talking about the number of hectares, the number of square miles of reef that we're restoring, right? So this is kind of the question, is there really a reef scale dent that can be made with these activities? So this was actually a question that my colleagues and I sort of tackled a few years ago. So we set out to sort of observe the entire reef and actually count all of the acropora colonies that were growing on a bunch of individual reefs. And we had actually done this back in 2006, shortly after the species were listed. And so then we were able to go and redo these surveys. So we used just a flag with a little GPS unit, and the snorkeler just kind of, you know, swam back and forth and back and forth. And every time you run into a colony, you hit a waypoint on the GPS, and then you can download those. And so we were able to compare the number of acropora on a given reef between 2006 and then 14 and 15. So we end up with something like this, right? So as you might imagine, a snorkeler asked to like cover all of the reefs swimming back and forth. It's actually not as easy as it sounds. And you can see like the snorkeler was working up here was a little different than the snorkeler was working down here, right? So this is not a perfect census method.
There was more current up there.
Exactly. This is not a perfect census method by any means, but it did enable us to try to look at this question at the full reef scale. So, and we felt that we had reasonable coverage. Again, this was in shallow water, and so, you know, the scale here was such that we felt like there was pretty good visual coverage of those areas. So this is Grecian rocks. Mm-hmm.
Great place. Is that during the die-off?
This is 2014, so it's a little tricky because some of the sites we actually surveyed before the bleaching, and then some we surveyed after, and that's why we went back and did all of them again in 2015, because we kind of had straddled that. Yeah, exactly. And so I don't recall exactly. I think that this was before the bleaching got bad anyway. So then we could put our pictures together from the 3 different years, and we kind of carved out a section of the reef where we had, you know, sometimes we had gaps at this end or that end, so we carved out on the map the area that we had common observations, and then we could calculate the total number of colonies that we had in each year. So for this example, for Grecian rocks in 2006, we counted 429 palmata colonies. Wow. Not bad, but only 39 Cercopornis colonies. There weren't that many. Now, in 2014, all right, we had a lot less palmata. We had exactly the same number of Cercopornis, oddly. But now look what happens in 15. So then after the effects of the first bleaching year, Again, the palmata tanked, okay? But what happened to cervicornis? It didn't tank. Guess why? There were 600 cultured outplants that were planted on Grecian rocks in that interval, all right? So look, that yielded a huge benefit, again, at the reef scale. So overall, when we surveyed like 10 or 12 reefs, some of them had outplanting effort placed on them, and some of them hadn't. So some was just the natural course of things. And so at the sites where there was no outplanting, we showed an overall decline of 40% in staghorn density and 70% in elkhorn density between 06 and 15. And again, the contrast in the sites with staghorn outplanting, there was a 13 times increase in density, okay, for cervicornis. So that was pretty cool. So again, if we look at the reef scale now, we had another bleaching event in 15, now we had a hurricane, you know, there is always something else to look at, but the point that we were really pleased about is that if you just go out and swim around the reef, you can really tell the difference. And that's what this study was showing.
Are there species that are bleaching resistant that we ought to be looking at?
There are a few. There's certainly variation amongst. So the question was about is there variation between species and how susceptible they are to the thermal bleaching? And there is some. Unfortunately, they're really like robust species, especially the bleaching like Prides asteroides. It's kind of robust to everything. The mustard hill coral, it's kind of fine. Nothing seems to bother it, but it's kind of a little runty, unexciting coral, and we don't really care about it. Unfortunately. Again, most of these, the large-sized corals that are really contribute to the reef structure, they're all quite susceptible. And susceptible also after bleaching to disease mortality that tends to follow on, and that's a really deadly mix.
To take that thought a little bit further, are there specific genomes in a crop or a palmata and cervicornis that are more resistant to bleaching and mortality?
Okay, so the question is, are there certain genotypes within the elkhorn species that may be more bleaching resistant? And the answer to that is yes, and that's what I'm going to talk about at the very end. So hold that thought, that's another good one. Okay, so I'm going to shift now to talk about the larval restoration side, right? So we talked a little bit about the effects that fragment outplanting can have. So we're going to talk about the larval side now. And I alluded a little bit in the beginning to these different steps that are involved, but if you think about the entire life history of a coral, it's pretty complicated. And again, for the larval restoration piece of it, we're actually encompassing the majority of the complicated steps in the life history. It are the ones that we're trying to provide sort of assistance and enhancement to all of these steps. So this means, you know, all through these steps. So including, this is just another sort of view of that drawn figure where you can see the actual embryo spawning. This is the embryo of 4 cells, right? Remember developmental biology from way back when? There's your little 4-cell zygote. A passive planktonic larva where it's just drifting around. By the time they look like this, they're actually swimming. You can kind of see this lighter area around the edge of the larva where it has cilia actually, and so it's able to swim around a little bit, has some directed movement. Eventually it'll develop the capacity, they start swimming down and start probing and searching for that special place on the reef that's going to be their forever home, and then sediment metamorphosis, etc. But so again, in terms of the restoration aspect of this, it's a matter of sort of providing support and enhancing each of those complicated steps. So that starts with predicting the nights that the corals are going to spawn. Remember I said in the beginning, they only spawn once or twice a year. And it happens to be in the middle of the night for the most part, which is kind of inconvenient. But we're out there in the middle of the night. You can see those little collectors that we use. From this picture, you can see the gamete bundles are spawned. So again, these bundles have egg and sperm packaged together. Here's a star coral. And because the eggs are mostly made of fat, because that's what obviously this is just like an egg yolk, they float. They're buoyant. So they'll float up into these collectors. We collect the gametes that way. And then go through a big process of fertilization stabilization, the larval culture, settling them onto artificial substrates, usually in a lab. You can see we've used lots of different types of dishes and tanks and aquaria and flow through things. Seacor's also worked a lot with workshops and really using these spawning opportunities to spread capacity. Many of our partners are from public zoos and aquaria and have been working for many, many years on these techniques, these culture techniques, and workshops are done every year at the Secor sites around the spawning event. So it provides lots of bodies to help with the culture activities, but also provides opportunities for capacity building. So again, in each of these stages, we have sort of assistive steps. We'll add these artificial substrates once the larvae are competent, they're developed enough that they're ready to settle. So through many years of experience and trial and error and research, these 4 steps we can perform pretty reliably, actually. The prediction, the spawn collecting, the culturing of the larvae, these are really pretty reliable steps. Now, this one about the survival and growth, after we put them back on the reef, this one we still have some challenges. But we have had some success. We really have, these are examples of Secor activities. These are both examples come from Curacao, one of our main research sites, where they have a lot more coral actually than we have here in Florida. They actually spawn like 3 or 4 times a year instead of once, so they're actually able to make a lot more progress there. But they have managed to, some of our larval, restored larval recruits have actually grown up at 4 years and have been absorbed spawning. So they've actually, those cultured, restored colonies have gone through that whole cycle. And this is an example for Colpophilia natans, the great brain coral, that also has had pretty good success with growing. This is a 4-year-old one. It shows you, right, the elkhorn coral in 4 years is big enough to spawn. The brain coral, not so much, right? So again, this shows you some of the difference between the species. But again, onesies and twosies, we have a few of these where they really are able to go through this entire process. The challenge really is getting it up from the onesies and twosies to this, again, this scale issue, same as we were talking about with the outplants.
Margaret, are you growing them the whole time in an aquarium setting?
Historically, generally we have. We've been growing them in the lab for the most part.
And so at what size then are you taking them out to the reef?
So it varies. Different locations will do it differently. You want to give them at least, the question, by the way, was about how long, if they were always raised in the lab, and how long they would stay in the lab before you would outplant them. And I would say at least you want to give them a couple of weeks after they've settled, because you want them to go through the full metamorphosis process and start laying down skeleton, because that's the only way you know they're really well attached to that substrate, whatever you're going to be outplanting them on. However, and I'll point to this at the end too, it may be one of the approaches to trying to improve their survivorship is to let them grow a little bit bigger before you put them out. So there's a lot of trade-offs involved there. And we don't necessarily have the best answer yet, but that's some of the stuff that we're at. But so this is the alternative now to those, you know, all that those laboratory tanks and small bowls and such, where we're now moving towards trying to put these large pools actually in situ, in ocean settings, and just dumping millions of larvae into each one. And then they have, again, their larval development phase of several days, 3 to 8 days or so, and then these design substrates. This is the other sort of piece of technology that Secor is really working on developing, putting these substrates in when they're ready to, they can go directly into the pools. And then again, the pools are designed so the walls will come off, and they can, again, be given a few more weeks, a few more months to grow and like undisturbed before they get placed out in the reef. So this is, again, these are very developmental stages here, but these pools and then these design substrates, the goals of these tetrapods, the idea, a lot of the cost of outplanting any type of coral, either larval settlers or fragments, is in the gluing step, right? Patty's nodding her head. The ladies probably glued a lot more corals. So the idea of these tetrapods is that they're self-stabilizing, right? That's the shape you see on like groins and jetties and stuff like that, right? Because those units settle down together and they don't need to be attached. They kind of self-stabilize. So the idea with these is, again, if we can settle them in these large numbers, in these large pools with lots of larvae, you can just kind of wedge them in. Don't need to glue them, don't need to nail them or cable tie them or anything. And they can stabilize on the reef. So again, the other goal with these design substrates, you see there's some grooves on here and some other textures. Our other goal with these is to provide those little microhabitat characteristics. They're going to help them survive better as well. So there's dual purpose here with the substrate engineering to improve both the logistics of putting them out there, but also the ecology of giving them good habitat to survive better.
What kind of habitats would you use for those types? Would they range from like sand and like hard rock bottom to coral reefs, or?
Let me show you some data,
Sure.
if I may. So the question was about what type of habitats the tetrapods would do well. You had another question though.
I was asking what the material was for the tetrapod.
So the current version that we've been testing are made of concrete, and again, a manual process. We have a couple molds and making them by hand. So again, part of our process right now is really working on some industrial engineering as well, industrial processes for industrial manufacture, but also looking at alternative materials like ceramic and glazed ceramic that may still provide good habitat for corals, but hopefully less habitat. You can see, so here's a few of them that were used in a pilot study in Curacao. You can see how they just kind of blend right in. Now, so this is some data. It was just recently published, I said, from sort of a proof of concept, a pilot study in Curacao, where in that case it was a different coral. It was a little, rinky-dink coral, like I said, we don't really care about, but we had larvae, and we figured the larvae, larvae are larvae at some level. So we were really testing the substrates, right? So this is not an exciting species, but what this is showing is sort of the yield of coral. So the number of tetrapods that we put out with coral settlers on them, and there were many settlers per, we put them out with more than one, but the number that had at least one live coral on it after a year was around 70%. And the cool thing too is that it was about the same after 6 and 12 months. So they kind of settled in and stabilized maybe around that time. Now you notice this. Then we put these out in different types of habitats. So again, really rugose habitats down to sort of flat, hard-bottomy places more. And not surprisingly, the flat, hard-bottom places had a lot lower retention, right? Not surprising. So, but this means that these tetrapod shapes probably are appropriate and useful in most reef habitats that have a decent bit of rugosity. So Patty's asking the size. They're big enough, again, then heavy enough. Part of the deal, they're hefty enough to kind of, again, stabilize themselves. Okay. So yeah, they're, I guess, like about 4 inches or so in dimension. So this, as I said, a pilot study we've been doing. We're currently working on sort of generation 2 of the tetrapods. We're engaging some engineers and some other folks with some different types of expertise, like I said, in terms of manufacture and that sort of thing, to try to get large numbers of these types of substrates ready for next summer. So, the protected growth, so the growth question, several folks had a question about that. And it does seem likely that, a coral this size is going to have a better chance surviving than, can you see that this is coral? Right, it's a couple little individual polyps there. So, this was work that I had done in collaboration with Moat Marine Lab. They have a really amazing land-based facility. They're incredibly skilled at growing corals on land in tanks. And so we had collected and gone through all those steps, settled a bunch of elkhorn settlers, and these are what they look like when they've been growing in the moat tanks for a year, and then these are the brand new ones. So these guys were born in 2013, essentially, and these were born in 2014. So, perhaps not surprisingly, when you put these out, actually, in the nursery, we hung these on some of the nursery trees in Moat's nursery, these guys dwindle away, and these guys get bigger. All right, so that shows you. So again, the idea is, though, that there's a trade-off in the investment that you make per propagule and how many you end up with, right? Which are really our goal. So I don't know if any of you studied ecology in college or whatever. This is a common concept about life history, strategy. I don't know if anybody took ecology. But, there are different kinds of organisms. So, like goldfish have 8,000 offspring a year, and apes only have 1, right? That's because they make a much bigger investment. And, humans have few. You make a lot more investment in each offspring. You get higher quality offspring and hopefully smarter and better able to survive, right? So, we have somewhat of the same concept at play here, where we have have trade-offs, they said, in the amount that we're going to invest in each individual larva or settler versus how many corals we're actually going to end up with on the far end that are actually going to contribute to the reef. And they said, we don't have the answer here. I have a couple of other examples. So I showed you the ones that grew out and then outplanted to the reef, and those are elkhorn coral that were doing pretty amazingly great. These are the tetrapods where, you know, we're settling them on the substrates, but then putting them out to the reef, well, sort of scattering them out on the reef and kind of letting them do their thing pretty rapidly. This is the other extreme. This is a recent effort in the Philippines where they actually kind of built this little cover over the reef, and they just shoved the larvae in there before they were settled. So that's kind of this concept underneath that cover there, exactly. So they laid out this cover, and it kind of sealed around the edges, and then they just injected the larvae while they were still swimming into that area so they would settle directly onto the reef. So that's kind of the extreme that we're talking about. So this is less investment, and this is, you know, 2 or 3 years worth of investment by the team down at Moat. So again, these are questions that we don't have all the answers to yet. These are some of our research foci. But I think it's likely that, you know, just like, you know, apes and goldfish don't necessarily live in the same environment. I think different reef environments and different contexts, the answer may be different. And so that's part of the challenge with us trying to optimize things, because the optimum may be different in different situations. Okay, one other thing I wanted to mention though, and this is something where SECOR really is moving forward very successfully, as I said, especially due to the efforts of our staff in Curacao, is obviously most of the effort, especially here in Florida, is focused on Eucroperas. There's good reasons for that. It's fast growing, it's an important species, they're endangered, etc. But, you know, 2 species do not a reef make, right? So we know that all of these other species are also important, as I said, in reef building, in all of these aspects. So we are really placing a lot more emphasis. I showed you some pictures of this guy earlier, the Colpophilia natans. These brain corals have had a lot of success with raising their settlers, and we're really investing a lot of effort in diversifying the species that we're working with, larval restoration. And part of the reason for that, again, this is kind of a hypothetical, but it corresponds with the observations they've made in Curacao. You know, all reefs, even existing reefs, are not the same, and some of them are in much worse shape than others. The elkhorn and catsagon coral really are kind of the wimps. You know, they're the most sensitive species actually on the reef in terms of, you know, salinity or temperature. I mean, pretty much any environmental parameter you mentioned, they're the most sensitive ones. So they really do require the healthiest habitats that are out there. And it may be that some of these other species, I said, these brain corals and some of these other species that have a wider habitat tolerance may also just be better suited for restoration in certain areas. And so this is part of our strategy as well, by incorporating these additional species, and as we, again, gain a little bit better ideas about how to optimize this, but really sort of match the species to some extent with the reef habitat where they're going. All right, last change of gears here. I'm gonna finish up pretty quickly, but we were talking there was a question earlier about is there variation in individuals of a single species to different types of stressors? And so that's the question I'm gonna address here at the end. I'm going to talk about disease first, though. Disease is still a factor that affects a lot of the outplants, as well as wild corals. As I alluded, they kind of behave the same, which is kind of what you want, except when they're dying. But disease, you can see, this is an example of some of the larger, these are outplants that Ken had done early on at Molasses Reef. So these we think are like maybe 6 to 7 years old at this stage. They're good size, these are spawning size corals. But you can see, these ones are all dead recently. And these ones are like half dead. All this white part is disease that's just kind of wiping through that reef. So it is a big concern both for outplanted corals and for wild corals. But we know that there was scientific literature from a study done in Panama, actually, that suggested that there were a few genotypes, a few genetic individuals in the stagmo population that were actually resistant to disease. I think it was like 6% of the genotypes that they looked at in Panama. But we hadn't really thought about this or looked for it in Florida. So we undertook a study to kind of do screening for this characteristic amongst the nursery genotypes, the nursery stocks here in Florida. So we worked with CRF here in Key Largo. We also tested a bunch of genotypes from the FWC nurseries down in Marathon and the University of Miami nurseries. And so the idea is a pretty simple-minded idea, but again, in the nursery, each individual has lots of fragments that are hanging on the same tree. So we can get 8 or 10 replicate fragments that are the same genetic individual. We can find an actively diseased fragment of coral and just like staple it right on there to see if it makes the other one sick. So in some cases, 7 days later, it's made it all sick and dead. And in some cases, here you can see the fragment's perfectly healthy and fine, and the sick one's dead, but the other one's perfectly fine. So this is a kind of straightforward type of assay that we can use to quantify that type of trait that could then be useful as we sort of design these restored populations. So in fact, when we did this, and so this is data from 2016, we tested 16 different genotypes of Cervicornis, of the saddlehorn coral, and 6 genotypes of elkhorn coral, and indeed we did find significant variation in the susceptibility of disease. So this is showing several zeros, so that means that none of the fragments that we exposed the disease to came down with disease in 7 days. Some of these genotypes had 80% of them that came down with disease in 7 days. So that's the type of variation that we see. But it wasn't just in the risk, so we call this the risk of transmission, the proportion of those fragments that were exposed that came down with signs of disease. But then for the ones that did come down with disease, if we look at the rate of tissue loss, so how fast that white scar is spreading across the fragment, we also saw a lot of variation. So some of them, even if they contracted the disease, it didn't bother them too much. They just had a little teeny bitty lesion, and some of them, as you saw, were completely dead. So there's actually sort of 2 parameters here where we're seeing potential variation, and that suggests, maybe, I think that's interesting, it suggests there may be different biological mechanisms by which they're resisting disease. And so there's this study is actually ongoing in the sense that these different genotypes now, we're actually doing the translation, we're assessing the transcriptome of these different genotypes to try to look at if there are certain genes that we can identify as accounting for this variation in susceptibility. And then if we can, then maybe that gives us something to actually sort of target, to tweak around, or use those to try to spread those genes throughout the population. So that's so question about bleaching variation then, and the answer is yes. So here are some examples. These are some of the other genotypes, Cervicornis genotypes from the CRF nursery. You can see these are 3 replicates of this U25. They're all pretty darn white. This was also in 2014 or 15, I can't remember. This one, this genotype, you can see all 3 of these, like, see how the tips are kind of white? So it had like a little bit of bleaching, but all 3 genotypes have the same pattern, or all 3 fragments of the one genotype show that same sort of partial bleaching pattern, and this one was totally fine. So again, we have this same type of variability for bleaching susceptibility that we can observe. This is just a random example from horseshoe reef. These are 2 Orbicella star corals that are, you can see, literally growing next to each other, seemingly the same environmental characteristics. This one's obviously really bleached in bad shape, and this one seems pretty much fine. So we see this type of variation in all sorts of corals. So the question is, what's the biological mechanism for that variation? And then how might we use that variation wisely in propagating those traits in our restored populations? Questions?
So the algae that's the inhabitant that's bleaching out is a factor in this as well. Are you looking at significant differences in the genotypes of the algae as well as of the corals?
Yeah, so the question is about the algal symbionts that are in the corals, because they do have a strong influence on the bleaching susceptibility of the bleaching process. So yes, and so in the star coral and these guys down here, it is quite possible. So this species hosts a wide range of different symbiont types, and particularly this D type is the one that's relatively bleaching susceptible. Now we did actually genotype all of these colonies in a horseshoe, and I don't remember for these particular colonies. It's likely in this case that the Zox types may have had an influence on that variation that we see. In Acropora, they're really not as variable in the types of Zox that they host. So they're pretty much all A. So we did a huge amount of sampling in 2014, and the 2014 bleaching of Palmata across a range of bleaching phenotypes. So in other words, colonies that bleached, colonies that didn't bleach, and then colonies that were half and half, right? Some they bleached on this side and not this side, because you see that a lot as well. So maybe, you know, there was different zooks on this side of the colony than that side. So we sampled within colonies in different bleaching phenotypes. We could not find any attribution of zooks type accounting for that variation in elkhorn coral. So in Orbicella, in the star corals, yes, that does tend to account for a lot of the variation in bleaching that we see as different types of zooks, but in Acropora, they just don't host as wide a variety, or they don't tend to. And so it's less, it's a safer bet that it's something about the coral animal itself that's variable in the case of Acropora. But yeah, no, that's a real good question. So as I said, so that's, I sort of closing down, I think this is really our challenge, is using this type of variability in a wise way. And what do I mean by a wise way? It doesn't mean that if we find one of those disease-tolerant or disease-resistant genotypes of staghorn coral in the UM nursery, then we're going to go out and just use that one for all of our restoration, right? That wouldn't be using it wisely. All right? But using it wisely might mean using that genotype, again, to cross with other genotypes in something like we would call selective breeding, right? We do this with lots of types of human endeavor. So that's a possibility. Selectively breeding the symbionts, like we were saying, the symbionts have a big influence on breeding. They also happen to have really short generation times, and they're easy to culture in the lab. So they're actually a pretty good candidate for artificial selection. If you want to think about that, if we take the zooks into the lab, and this is some of the research that's going on right now in Australia, take different zooks types into the lab and just keep them hot for 3 years. They go through about 40 generations, and sure enough, you end up with a strain that has like 2 degrees higher tolerance than you started with. So these